Chapter 3, Basic Principles and Concepts, Immunology and Pathology, Innate Defense Mechanisms, Part 2. The first lecture of this section we discussed the different proteins and things that would be found as part of our first line of defense for something that gets in us or on us. Complement, immunoglobulins, lysozymes, and whatnot. Here in this lecture, we're going to be discussing what comes about when the cells of the innate immune response come into play. Turns out many of these cells, what they're going to do is what it shows right here. They're going to phagocytize. Phagocytosis literally is a feeding mechanism that is found in single-cell protozoa. Single-cell protist, like an amoeba, will come grab a hold of something that it wants to eat or wants to take in as a food particle, and then the rest of the plasma membrane just wraps around. Think of it as a giant bear hug. A giant bear hug where the plasma membrane pushes out, around, and encircles. Here, when it encircles, this part fuses together and becomes a new plasma membrane, leaving whatever that particle was, a bacteria, some other smaller cell, whatever, leaving it in a large vacuole. Well, turns out some of our immune cells do the same thing. Right now in your body, you have macrophages that are on patrol underneath the epidermis. They're swimming through the dermal layer. They're swimming through your muscles. They're swimming through your different organs, looking for anything not you. If they find something not you, they're going to use, like we talked about last lecture, either a scavenger receptor, complement receptor, or one of the PRR receptors. They're going to grab whatever's not you, envelop around it, and take it in. That is phagocytosis. Okay? It is a means of eating, a means of taking up things smaller than that cell. Bacteria, virus, cellular debris, parts of parasitic worm debris. When the cell's phagocytized, they put whatever that particle is, the bacteria, the virus, whatever, into a vacuole. Okay? That vacuole is basically just a lipid bubble inside the much bigger cell. Nothing happens to the bacteria, the virus, the cellular debris, whatever, until a lysosome, another smaller little lipid bubble made by the cells, but inside those lysosomes is going to be stored a bunch of enzymes, a bunch of digestive enzymes. There's also going to be quite a few different types of reactive oxygen species and reactive nitrogen species. Got to remember, an oxygen-free radical, or some oxygen reactive oxygen species, H2O2, O2 with that little negative, things like that. What they are going to do, those reactive oxygen species are going to go and strip hydrogens off of everything, anything they come into contact with, proteins, lipids, DNA. So it's going to start damaging the bacteria, the virus, whatever, from the inside out. The enzymes, the proteases, the lipases, the DNases, the RNases, and whatnot, are going to go through and start basically cutting up whatever that cell or that cell debris is. When it comes to invertebrates, okay, we got to remember invertebrates are going to have their own version of some innate cells that are on patrol. These are going to be specialized cells that are looking for anything not self. They're going to either go and, take it up via phagocytosis. If they can't do that, they're going to wall it off by more and more of the cells just glomming on to whatever has gotten inside the invertebrate body. As more and more of these cells, these innate immune cells, glom onto it, they start to form a thicker and thicker layers of cells that are now going to become tissues. They're going to wall off that little bitty bacteria, that little bitty virus, that little bitty parasite. So it's now walled off from the rest of the invertebrate body. This is also going to help in wound repair, because you got to remember, a wound, whether it's in an invert or us, a wound is going to damage tissues. It's also going to damage cells. Those damaged cells don't repair themselves. Those damaged cells just die, and they need to be removed. When it comes to invertebrates, we refer to these as you see right here: archaeocytes, amoebocytes, hemocytes, coelomocytes. I mean, look at their names. Amoebocytes. Okay, I just got done talking in the last slide or so about how, single-cell protists, the amoeba, go and swim and grab hold of, well, when they saw these cells in whatever, invertebrate they were studying, it reminded them of amoeba. So, hence, amoebocytes, amoeba cells. Amoeba-like cells, I should properly say. Coelomites, coelomocytes, I guess, would be the correct pronunciation. These are going to be cells that literally are swimming around the interior cavities of whatever the invertebrate is, the insect, the slug, the snail, looking for anything not self. But when it comes to us vertebrates, when you look down here, you'll notice right there in the middle, stem cells. Okay, we start with stem cells. Those multi-pluripotent stem cells are not the true embryonic, I haven't learned anything yet, total blank cells. They've been told a few things. They've started on a pathway, but they're still blank enough slates that they can become pretty much anything. Some of those cells are, they're going to go split and go down different lineages. Some of them are going to start a lineage of myeloid. These myeloid cells are going to become pretty much everything we think of as white blood cells. Here in the figure right there on the far left, it shows the different developmental stages, the maturation stages, I should say, of a cell as it goes and becomes the monocyte in the blood, leaves the blood, enters in the tissues, and becomes a macrophage. You would see some of the same things for neutrophils, eosinophils, basophils, and whatnot. The other lineage is going to become the lymphocytes. Okay, these are cells that we associate with the lymphoid organs, the ones below the jaw, the armpits, the groins, all through the mesentery in our abdominal cavity. These cells are going to develop, as we see, into what we call the B cells and the T cells. Okay, B cells, antibody-making cells, okay, specifically known as plasma cells. Some of those plasma cells will then further differentiate, become memory B cells, so that if you're ever exposed to that antigen, that invading organism ever again, you can start making antibodies within a day or so, all over again. The T cells we're going to see, helper, cytotoxic, and whatnot. They are direct killers themselves, or they help regulate all the other cells you see there. So keep that in mind. When you're talking innate as well as adaptive, everything starts from the stem cells that will split. They will split. The innate, these are the ones that are going to become the macrophages, the neutrophils, the eosinophils, the basophils, and as we're going to see in the next set of lectures, the adaptive, the B cells, the T cells. Now when it comes to us vertebrates, we're going to find that the cells, even though they may be the innate cells of the myeloid, development, the macrophages, neutrophils, things like that, that the cells will take on different roles than we think that is normally attributed to them, I should say. They will take on these different roles based on their location, based on what tissues is housing them at the moment. Is this just a transient passing through, or are they being housed there? You take, say, like the Kupffer cells, macrophages that are being housed in the liver. Okay, their job is to help clear out damaged red blood cells from the blood that's flowing through the liver. That's their job. They phagocytize, digest it away. The microglial cells, you're going to find this in the central nervous system, looking for anything not you that may have made it, through the blood-brain barrier, through the meninges and everything, into the central nervous system itself. They're going to find it, they're going to kill it, or they're going to go and find, they're looking for damaged neurons, damaged Schwann cells, damaged whatever. Their job is to clear it out. You'd also take into account we utilize dendritic cells. Dendritic cells are the intermediates between the adaptive and the innate. Okay? The adaptive and the innate don't always talk together so much. But you have a dendritic cell that's going to take from the innate any antigens, anything they may have found not you, and then will deliver it to the adaptive cells, the B cells, the T cells, saying, here, have you ever seen this before? Do you want to learn this? Then you have, granulocytes. These are the eosinophils, the basophils, the mast cells. These are cells that you're going to find in vertebrates that have developed this ability to overproduce certain enzymes and certain digestive enzymes is what they've learned to overproduce. And instead of just excreting them out willy-nilly, they will take them and they will store them as larger and larger clumps of proteins that start to take on a crystalline structure under a microscope that makes the cells look granular, hence the name, granulocytes.